Hello everyone, welcome back to Statisticians Hub. Today we are delving into the world of t-tests in SPSS, a powerful tool for comparing means. If you are ready to unlock the secrets of statistical significance, you are in the right place. I am Shubham Roy and let's start the video. So what is t-test? T-tests are statistical hypothesis tests that we use to analyze one or two sample means. It is often used in hypothesis testing to determine whether a process actually has an effect on the population of interest or whether two groups are different from one another, just due to chance. T-tests are used for different purposes. In this video, we are concentrating on different types of t-test for means. The first t-test is student t-test or one sample t-test, which compares if the mean value of a population is equal to a fixed hypothesized value. The second t-test is two independent sample t-test. It is used to test the hypothesis if the mean value of two different populations are identical. The third t-test is to compare the average value of the same variable before and after any event, popularly called as the paired t-test. Now here we have our first one sample t-test. It is used to determine if the mean of a single sample is significantly different from a known or hypothesized population mean. Here it is the formula for one sample t-test: t equal to x bar minus mu naught by s by root n, and x bar is the mean and here is the s square, and degrees of freedom is as usual n minus 1. Now here we have some assumptions. Number one is the parent population from which the sample is drawn is have to be normal, and number two, the sample observations are independent, and the population standard deviation is known to us. Now here we have an example by which we can understood it perfectly. Here we have a random sample of 10 boys and these are the IQs of that 10 boys. Now we need to see that do these data support the assumption of a population mean IQ of 100. That means we need to find that whether these boys came from a population where the mean IQ is 100. So let's go to SPSS and find the results. Now let's start in SPSS. Here we have the IQ values and in variable view we already put IQ as the variable, which is a scale type of measure, and here we have the data. So to perform one simple t-test we have to go analyze, then compare means. Here we have one simple t-test. Click here. Here in first view you saw that IQ is there in the variable chart. From here we need to put it in test variable because we need to test for IQ, and the test value is 100. I put it as 100 because see in the question we saw that we have to find that whether this value, this sample is came from a population which mean is 100. See here. So that's why we put the test value as 100. So now just click OK, and here we have the result. In the first table we have the descriptives, and in the second table we have our important values. This is our p value, which is the most important thing, the most important value. So this value we saw that is p value is greater than 0.05. That means we accept our null hypothesis. Whenever p value is greater than 0.05 we accept our null hypothesis, otherwise we reject it. So in that case, what is our null hypothesis? Our null hypothesis is for one sample t-test there is no significant difference between the population mean and specified mean. So we accept this null hypothesis. Also we can find the answer from this t value also. If you do the t-test by pen paper, then this is a important value for you, but for that you need to see the t chart, and here for 9 degrees of freedom there is a critical value. So that's the other thing. In any other video someday we will talk about that, but for now this is our important value, the p value. By this we can interpret the answer. So that's all about the one sample t-test. Let's move on the next part. Now let's go to our next test, which is independent sample t-test, or called independent t-test. Now the independent sample t-test is used when you want to compare the means of two independent groups to determine if there is a statistically significant difference between them. Like in experimental studies, researchers use the independent sample t-test to compare the means of two groups subjected to different treatments. For example, a pharmaceutical company might use this test to compare the effectiveness of two different drugs. So here is the definition. It is used to compare two sample means from unrelated groups. Like for example, you can also take in a population we have male and female group. So we want to find something, or we want to test between these two groups, male and female. Then also we can use this. So this is the formula for independent sample t-test, equal to x bar minus y bar by s root over 1 by n1 plus 1 by n2. And these are the values of x bar and y bar, normal mean, and n1 and n2, you know, the number of samples or number of individuals in n1, the group 1 and group 2. And this is the formula for s, and the degrees of freedom is n1 plus n2 minus 2. So here also we have some assumptions to use this. Number one is the parent population from which the sample is drawn is normal. Number two, the sample observations are independent. And the last one is the population variance are known but have to be equal. Here we have one example to understand is better. So here we need to test that is there a significant difference in cholesterol levels between individuals who followed a diet intervention and those who followed an exercise intervention. That means here, as I said previously, here we have two different groups who followed diet and one group followed exercise. So in this example, we put 1 for diet and 2 for exercise, and these are the cholesterol values, and here we have the intervention. 1 for diet, that means this person follows diet and this person follows exercise. So now let's go to SPSS and let's find what gives us after the test. Now here we have the values of cholesterol and have the values of intervention, and in variable view we already put the variables. And now to analyze, we have to go to analyze and compare means, then independent samples t-test, and click. Here in the variable box, at first it is going to see like this, and from this variable list we have to put cholesterol in the test variable because we have to test for cholesterol, and intervention is our grouping variable because there are two groups in interventions. One is exercise and other one is diet. So we have to define groups. So group 1 is 1 and group 2 is 2. Now continue, and then okay. In the output table, here we have the values. This is the simple descriptives, and in this table we have to see this one, t-test for equality of means, because we are testing for equality of means. So here this is the most important thing, significance two-tailed, and in this significance two-tailed we have to see this value, because in the assumption we already see that for independent sample t-test there is assumption that we have to, the variances have to be equal. So here equal variance assumed, and this is equal variance not assumed. So we have to see this one, equal variance assumed, and this value is 0.046. So 0.046 means it is less than 0.05. So here we need to reject our null hypothesis. That means there is significant difference between the two groups. So in this way, we are going to analyze and interpret the independent sample t-test. Now, last but not least, we have the paired t-test. This one is for related groups, like measuring the effectiveness of a treatment by comparing the same individuals before and after. The paired t-test helps us see if there is a significant difference in paired observation. Like if there is a before-after case, you can use paired t-test. It is used when the two samples are equal in size and are not independent of each other. So here is the formula for paired t-test, and these are the values of x square, d bar, and degree of freedom is n minus 1. Now here we have an example. Like I said previously, here you see that we have to find and we have the iron contents of fruits before and after applying farmyard manure was observed as follows. So before applying farmyard manure, these are the values of iron contents, and after applying the treatment, these are the values. Now we have to find that is there any significant difference between these two. So let's go to SPSS and let's find. Now here we have the values before applying and after applying. So I already put the variables here, and in data we have the data. So first we have to go analyze, compare means, then paired sample t-test. Here we have to put before applying in this variable 1 and after applying in this variable 2 in this manner. First you will look this like this, and after that select before applying and in variable 1 and after applying in variable 2, and this is our pair. Now press OK, and in the output table we get the result. These are the descriptives, and we have to see this table, this last one. So see here the significance two-tailed value. This value is our important one. So this is 0.354, and which is much more greater than 0.05. So that means we accept our null hypothesis. This is the most important value. As this is greater than 0.05, that means we accept our null hypothesis, which is there is no significant difference between the two groups. That means there is no significant difference between before applying and after applying. So this is our interpretation and result. In this way we can analyze and interpret paired t-test. So I think and I hope all of you like this video and the tests. Thank you so much. And here this is the end of our analysis part for today. I hope all of you enjoyed the video. If you like the video, then please like the video and share, and subscribe the channel Statisticians Hub for more this type of videos. Thank you so much.